Julius Lux, Associate Professor of Chemical and Biological Engineering at Northwestern, to introduce our next speaker.
Yes, thanks everybody. It's my great pleasure to introduce my good friend and colleague Peng Yin. Peng is a professor of systems biology at Harvard Medical School and also a core faculty member at the Wyss Institute at Harvard. His lab is pioneering in nucleic acid nanotechnologies, both from the architectural standpoint, building interesting objects with nucleic acids, as well as dynamic control, in particular pioneering work on controlling cellular behavior. He is a recipient of an incredible number of awards, including 3 from the NIH Director's Office, the New Innovator Award, Transformative Research Award, and a Pioneer Award. He's also received the NSF Career Award, a DARPA Young Faculty Award, ONR Young Investigator Program Award, NSF Expedition and Computing Award, the first ACS Synthetic Biology Young Investigator Award, a World Economic Forum Young Scientist Award, and a Tulip Award for DNA computing and molecular programming. He's got a varied background, graduating from Peking University with a BS in biochemistry and molecular biology and a Bachelor of Economics. Then went on to do an MS in molecular cancer biology at Duke, and then completing his PhD at Duke, but in computer science, and then his postdoctoral training at Caltech. He's also got an entrepreneurial streak as co-founder of 3 companies, including UltraView, NewProbe Global, and Taurus Biosystems. So welcome, Peng. We're looking forward to hearing what you have to tell us.
Great. Thanks so much, Julius, for the really wonderful introduction. Let me share my screen here. Let's see. Oh, can people see my talk?
Yes.
Yes.
Okay, thank you. Thanks again, Julius, for the really wonderful introduction, and thanks for organizer for the invitation. It's my great pleasure to be here and to visit Northwestern University virtually. I've been on campus physical 3rd time, but this is the first time I think for the virtual visit, and I share some new work here. So as Julius mentioned, so our lab is interested in engineering nanostructures primarily using DNA as an engineerable material. So here we're considering DNA and also RNA as the physical material rather than a genetic material. And so importantly, the DNA strands are information encoding material, right? So the DNA strands really carries information, and which are essentially the sequential arrangement of the ATCG4 basic units. The remarkable thing about DNA strands is that when you specify the digital information encoded in DNA strands, that is, when you specify the sequence of DNA strands, then you have opportunity to engineer them into nanostructures that with user-prescribed molecular scale behavior, geometry, dynamics, and other interesting properties. So for today's talk, I'm going to first share with you some of our work on how to engineer DNA nanostructures and nano devices. And then I'm going to share some work on the application side, in particular, how to use DNA nano probes to advance aspects of molecular imaging. And finally, if time allows, I'm going to share some recent work that we just starting now to develop a single molecule protein fingerprinting or protein sequencing using DNA-based imaging platforms. Programming DNA. So here there are, I think, 3 aspects I want to share here. One is how do we engineer DNA-based nanostructures with a user-prescribed geometrical property, and then next dynamic self-assembly and disassembly, and finally some dynamic synthesis work. So geometry. So here, the field has developed a number of ways to engineer the complex structures using DNA strands. And the one particular motif that we have been working on, what we have been working with, is called DNA break. So DNA break is simply a single strand DNA, in this case for two-dimensional structure engineering, and the DNA break is a 42-base single strand. And then it can be conceptualized as a simple tau Lego break, And two DNA breaks can be engineered to bind to each other if they have engineered complementary binding domains. So then by simply engineering the domain-to-domain complementarity of DNA breaks, then you can design the DNA strand to assemble into complex molecular scale structures. For example, we were able to demonstrate the construction of 100 different two-dimensional structures. Each structure here contains hundreds of DNA strands, and each one has a unique user-prescribed shape on the 100 nanometer scale. And later, in collaboration with my colleague at Wyss Institute, William Shea, we were able to extend the 2D DNA break structure architecture into 3D-dimensional construction. And so shown here, 100 different shaped DNA nanoparticles. And the scale bar here is 100, and these particles microscopic 25 nanometer in diameter. So tiny structures, but we can encode the complex geometry in the structures. For example, this particular particle here has 3 parallel holes, and from the electron micrograph, you can see the 3 holes quite clearly. And this guy has 6 perpendicular holes, and again, you can see this 1, 2, 3 quite clearly. So in the sense that really the DNA breaks can be thought of as a nanoscale 3D nanoprinter, whereby merely specifying the digital information of the sequence while encoding the breaks, you can manufacture complex 2D and 3D three-dimensional particles. So this is the first generation DNA break, and then later we were able to further expand the concept, and with the second generation DNA break with even longer encoding domains and binding strands, and we were able to engineer structures that contains, instead of hundreds of unique components, here we scale up capacity to engineer structures that contains tens of thousands of unique components. For example, here, and by designing 30,000 different DNA breaks, and each one designed to fit in particular voxel location in the Q-bound structure, we were able to engineer these 30,000 different strands to self-assemble into user-prescribed structures. And shown here is one structure we demonstrated. And this is both I think artistic rendering, but also actually physical model, since that every single brick here represents physically the physical position of a DNA brick. So the structure is a billion times smaller than what I'm showing here. So from outside, you can see the cuboid shape, but inside there's a user-defined teddy bear-shaped cavity. So in addition to engineering DNA strands to self-assemble into multi-component system, and a similar principle, that is a user-prescribed complementarity-driven hybridization, can be used to engineer a single-strand long polymer DNA strand or RNA strand to self-fold into complex architectures. And this in collaboration with Wyss lab at Arizona State University. And so, for example, this is one of the largest structure we demonstrated, and here we're looking at one single strand of DNA containing 10,000 bases, and it can be engineered for back and forth into this particular shape. So these structures are not only the interesting from fundamental perspective, but they can also be practically meaningful and useful. And the one perspective is that one application angle is that these structures can be modified with other functionally relevant moieties with nanometer precision. For example, by decorating the DNA nanorod with a particular combination of fluorophores, and we were able to engineer the DNA nanorods with more than 200 different optical signatures. And in another work, we engineered more compact DNA structures with a digitally prescribable and decoration of a control number of fluorophores, and hence you can have a programmable color and intensity. And this kind of a structure could be useful for highly multiplex single molecule tracking. So in addition to structure, and we also have been doing some work in programming dynamics. And one particular motif we use is this so-called total break. It's essentially DNA hairpin structure which contains 3 domains A, B, C. Importantly, the hairpin structure has exposed a single strand region known to the field as a toehold. Now, a complementary trigger sequence A star can bind to A, where a process known as toehold midi branch migration, where A star fully binds to A, and this will open up the hairpin. Importantly, it will expose the initially hidden toeholds on B and C. Now, B and C with their newly exposed toeholds can serve as triggers for other suitably defined hairpins. For example, now B can trigger the opening of this hairpin with a B star sequence. So without going into details, and what we are presenting here, essentially, when you step back and we are looking at two chemical species, 2 molecules, H and H2, initially they are engineered to be metastable. Only when you introduce another particular molecule T, when T can assemble spectrums H and then activate H, then the activate the warning H, and then can assemble with H2 and activate H2. Then without getting details, and this provide a platform for us to engineer programmable kinetic cascades of hairpin opening and concurrent assembly and disassembly. And this is this provide a platform to engineer rather complex synthetic molecular behavior, including catalytic circuits and the locomotion of the DNA fragment along self-assembled DNA track, also trigger dendritic growth. As a fundamental principle that can be demonstrated both in test tubes in the ideal environment, but the principle can also be adapted into living cells, this time using RNA rather than DNA, as RNA can be easily transcribed in the cell. So as one demonstration in collaboration with my colleague Jim Collins and technical postdoc Adam Green, jointly advised by Jim and me, and they engineered the molecular the structure called the toehold switch. The toehold switch is a single-strand RNA structure which encodes of gene of interest. When you transcribe the RNA hairpin and the toehold switch in the cell, initially the ribosome binding site is hidden and as you just coded. As a consequence, the encoded gene translation is repressed. However, when the toehold switch encounters another trigger and trigger a single-strand RNA T with the user prescribed sequence A star B star, then again through toehold midi branch migration, then T can bind to the hairpin, and then this will open up the hairpin, exposing the initial hidden RBS ribosome binding site and start codon. And the ribosome can be engaged and the translation gene can be turned on. So a simple device by implements a quite general molecular logic. If detecting gene A, then produce protein B. You can see that the protein encoding region and the trigger region are intentionally designed to be sequence dependent. So this make it the wiring of input to the output to be quite arbitrary. So again, without getting into details, and such programmable system can be engineered to be quite complex. And in a follow-up paper, we demonstrate that in the living cell, again, E. coli, the following molecular information processing can be engineered if detecting RNA input A1 and B1, but not A1 complement, or A2 and B2, and not B2 complement, and so on and so forth. If this 12 input and the RNA profile evaluation is turned out to be true, only then you turn protection of protein X. So in addition to the geometry and dynamic assembly and the disassembly, another aspect is to manipulate not only the component aggregation and disaggregation, but also to program the DNA synthesis itself. And this is primarily we call the primary exchange reaction. And so this is a very simple molecular operation where a catalytic hairpin can extend a primer A to be AB, right? A to AB. And a little bit detail here, as there's a particular reaction are going to be used for other imaging-based application. So here, we start with a primer A, and then A can bind to the catalytic hairpin, and here where A bind to A star, right? So just a simple hybridization between two complementary fragments. Now, when A binds to A star, the polymerase will be engaged, and the polymerase can extend A by copying B star and displace B. So it can extend A to be AB. Importantly, when it hits the following the position labeled by the black dots, and this is the chemically chemical distinct site which can stop the polymerization, and the polymerase will be disengaged. Now the newly synthesized B fragment is identical to the existing B fragment, so they can have a molecular competition, and eventually the AB can fall from the hairpin. Now A is turned to AB, and the hairpin is released, so it can catalyze, and the single hairpin can catalyze extension from multiple A species to be AB. And now if you have a group of hairpins with a suitable design sequence, and you can set up molecular cascade where autonomously and A can be extended to be AB, to ABC, and A to be A, B, C, D, E, F. And this this animation to show the process. So the hairpin is engaged, and you synthesize the first magenta fragment. Then through branch aggregation, eventually the hairpin will fall from the strand. Another hairpin is engaged, and then autonomously, and the strand can be extended to use a prescribed structure and sequence. And so such dynamic autonomous assembly can be coupled with other environmental signals to implement a number of conceptual applications. And these are not real applications yet, just shows a conceptual capability. For example, we demonstrate that the detection of microRNA signal can trigger the synthesis of a long concatemer, and which can then bind to fluorescent molecule and turn it to be bright. And so this can be single application and the possibility. And in another conceptual demonstration, if you detect a microRNA signal, and this input signal can trigger the synthesis of a DNA enzyme, which can specifically can trigger the synthesis RNA enzyme, which can specifically cut a target RNA strand to fragments. So these are the more fundamental aspect of the lab's work, where again, by programming the digital information encoding DNA and RNA strands, we demonstrate the ability to engineer complex user prescribed molecular level behavior, structure, geometry, dynamics. Such digitally programmable molecular systems with a nanometer precision and offers opportunity to develop diverse applications. If thoughtfully guided, and such application could be transformative for different application field. And so next, I'm going to focus on some work I've been doing on engineering DNA probes for advanced aspects of molecular imaging. In particular, I want to share with you how we can improve molecular resolution for the imaging using technology called DNA print, and how we can achieve highly multiple imaging using technology we call DNA exchange, and how to achieve signification for high throughput imaging using SIBR. And finally, I want to share with you a sequencing-based DNA nanoscope for mapping molecular scale arrangement of nanostructures. So resolution DNA print. So here, and the technology is quite simple. The idea is the following. So suppose we want to visualize a nanoscale object, in this case, and a synthetic DNA nanorods, and we can decorate this nanorod with DNA fragments that we call docking strands. In the solution, we introduce 4 of our labeled image strands, which are designed to be complementary to docking strand. So then the image strands will specifically bind to docking strand. However, the complementary binding is engineered to be sufficiently weak. And as a consequence, the binding is transient. That is, the image strand will bind here, and after a programmed residential time, and the image strand will fall off. Now, if we position the nanostructure to be imaged in the focal plane of the microscope, only when the image strand transiently binds to docking strand, this fluorophore will become apparently bright. So what you will see under microscope is the stochastic appearance and disappearance of these blinking dots, where the appearance of each blinking dot correspond to the transient immobilization of a fluorophore labeled image strand to docking strand. So we can control or engineer the blinking duration and frequency by independently engineer the binding strengths and controlling the imager concentration. Then if done in the right fashion, the blinking spots can be sequentially localized with a nanometer precision, and this will turn the fuzzy optical image into super sharp image. And of course, this not only applies to synthetic DNA nanostructure benchmarks, but also can be applied to biological systems. And here, using DNA modified antibody, which binds, in this case, some protein interest, and we were able to demonstrate, in this case, the visualization microtubules in fixed HeLa cells by applying DNA fluorophore modified DNA imager strands and enable the transient blinking, then this microtubule image can be turned from fuzzy image into super sharp image. So compared with other complementary super resolution imaging technology, for example, Storm and Palm, which uses special laser setup and special fluorophore to be turned on and off using the laser setup. And here we achieve the massive blinking for localization with super resolution imaging by simply engineering the transient molecular binding between a docking strand and image strand. So this makes the technology highly accessible and widely adoptable. But then importantly, as we do not label any particular target stable in the fluorophore, we only label it with a DNA docking station. As a consequence, in our skin, we do not suffer a phenomenon known as photobleaching of a single fluorophores. So because if any particular fluorophore is bleached, there's a larger reservoir of intact fluorophore labeled image strand can come here and to be replenished. As a consequence, we have no photobleaching, and this coupled with a programmable on-off binding and blinking enable us to achieve ultra high resolution imaging of densely packed molecular targets. So without getting details, Minjie Dai, a very talented student and later postdoc in the lab, were able to demonstrate the realization of a densely packed DNA strands on DNA origami structure, where two targets are separated by each other by nearly 5 nanometers. Right? So this is raw image, and this after automatic computer fitting. So you can see that Minjie can resolve individual points from each other in a digitally clear fashion. And then 5 nanometer is not only for technology showcase, but also is biologically meaningful, as a 5 nanometer is a size of small protein. From this optical image perspective, now it has opportunity to potentially visualize individual proteins on the surface cluster, and of course, provided they can be labeled and modified with DNA strands. So the next multiplexing. So here, and we want to share with you a technology called DNA exchange. So when people talk about multiplex fluorescent imaging, in the current I think scientific and industry world, really we are talking about practically 3 or 4 colors are really good, maybe 5, 6 colors. But that's not sufficient for unrivaled biological complexity. Really, we want to visualize structures and components not only by 4 or 5, but rather 10, 20, hundreds of species. And so when we overcome the so-called spectral overlapping and multiplexing limit in the fluorescent optical imaging, is to take advantage of DNA labeled and using DNA labels for engineer or certain barcodes and sequential imaging and capability. So in the exchange point, in the exchange method, if you want to visualize a large number of distinct molecular species, we label them with distinct molecular barcodes or DNA barcodes with orthogonal sequences. Then you introduce P1 star or fluorophore label P1 star image strand first, which will specifically only interact with P1, but not productively with P2 or Pn. As a consequence, you can specifically visualize P1 label targets and assign the pseudocolor. And importantly, we achieve such specific interaction and identification and localization P1 using transient interaction between P1 star and P1. So this is a practical meaningful because after visualizing P1, we can through a simple buffer change within minutes, wash away P1 star and introduce P2 star, which then can interact with P2 and visualize P2. And so on and so forth, then you can visualize a large number of molecular and species and through fast imaging cycles, exchange cycles. In the first paper published, we demonstrated the high resolution imaging of 10 synthetic nanopatterns and also 4 different protein components in the fixed cell. And later, we extended the capability to demonstrate the 8-color imaging of different protein structures in neuronal cells with a high resolution and high multiplexing. Now, the general concept in exchange is quite general. It's not restricted to the particular DNA print the platform, and we later extended generalized technology to be compatible with other super resolution and also standard resolution imaging platforms. And so, for example, this 7-color confocal tissue imaging, where we are able to visualize different structures, protein biomarkers in tissue sections. So technology validated to image both protein, RNA, and DNA. And now, so coupled with a very high resolution down to 5 nanometer or true molecular resolution, and with spectrally unlimited multiplexing, if we put these 2 capabilities together, and the platform offers exciting prospect of potentially turning such a cartoonish depiction of molecular structures of and what people think what cell look like into actual data, where we could imagine to visualize individual different molecular species in their in situ environment in the cell, and to build the so-called molecular atlas cell. And our current effort focus on, rather than protein, actually focus our effort more to visualize the chromosome. And this is a really fascinating structure, the 2-meter fiber information encoding fiber folded in the less than micron 3D space. So here is a visualization of the segment of a human chromosome number 8, and these are labeled with 100 kilobase molecular genetic resolution and with optical resolution of 10 nanometer on the lateral dimension and 25 nanometer on the vertical dimension. I think this is probably still the highest resolution in situ visualization of a chromosome to date. Then in addition to molecular imaging, and we're also interested in tissue imaging. So here, rather than building a molecular atlas cell, and the larger vision is a so-called building a cellular atlas of the body. And when we're thinking about tissue scale imaging to visualize a cellular identity rather than molecular identity, and the technical consideration becomes a little bit different. So if we want to build a molecular atlas cell, we want to achieve simultaneously very high resolution down to molecular scale 5 nanometer and very high multiplexing. When we consider building a cellular atlas body, then the most pressing need is rather than achieving super high resolution, but is to achieve very high throughput combined with high multiplexing. And to enable the simultaneous achievement of high multiplexing and high throughput, and we invented our technology called SIGR, signal amplification by exchange reaction. And this is built on top of our DNA exchange paradigm, but introduce signal amplification that are compatible with spatially unlimited multiplexing. So suppose we want to visualize biomarkers in the tissue or multiple protein species. And what we do here is we use a DNA barcoded primary antibody, and in one single staining step to label all the interested and protein targets with DNA barcoded primary antibody. And this is one slow step, like primary antibody staining. And now then in the second step, we use a SIGR technology to introduce to extend the short primer into long concatemer. Each concatemer have tens to hundreds of binding sites for imager species. Then we introduce imager species 1 or 3 times, 3 space at a time, where the imager species P1 star will specifically aggregate onto the P1 concatemer. And this will enable the realization of the target T1, but with many fluorophores and hence very high signal to enable highly sensitive detection, but also high throughput imaging acquisition. Then we wash away the P1 star, introduce P2 star with a T2, and so on and so forth. And this way, we can achieve simultaneously high throughput and high multiplexing. To synthesize a polymer and as a long concatemer, we utilize a particular implementation of the primary chain reaction that we described earlier. So in this particular implementation of primary chain reaction, we use a catalytic hairpin with A star and a segment. Then the A primer can bind to A star, then be extended to be AA, and then the hairpin can fall from the AA, and then the newly extended AA can be extended to AAA. And in this process, using single catalytic hairpin species, we can synthesize long concateners, and with a user-controlled length by controlling a number of chemical factors. So now these long concateners can be introduced to serve as scaffolds to aggregate imager labeled fluorophores, and using such approach. And then later, using we can also, in addition to linear structure, we can also engineer bunch structure, and we were able to demonstrate amplification signal in this case and to 188-fold, and this is comparable to the industry standard TSA amplification. So the utility of signal amplification is not only for sensitive detection in tissues, but also when we are talking about a larger scale tissue imaging. And with a fast exchange cycle, then the key bottleneck for imaging acquisition time actually becomes the imaging acquisition time for every single frame. Now, with the acquisition time for each single frame of image of a single frame image is fundamentally determined by the photon counts in that image. So when we amplify signal to be tens or hundreds times more brighter, then the image acquisition time can be reduced by 10 or 100-fold. So in this demonstration using FFPE tissue and using as a sleep amplification, we were able to acquire horizontal image and with millisecond, single-digit millisecond time scales for 100 micron field of view. Now, this is first validated with higher multiplexing, where we're able to visualize 7 different markers in the FFT tissue. And then this can also be applied to the cross section as exemplified here, 10 color imaging. So, right. So the technology is adaptable from protein and also to visualize nucleic acids. And without getting details, we are able to, using the so-called superfish technology, to visualize 7 different RNA markers in the cross section. So we set up 2 websites to widely disseminate as a protocol and capacity, superfish and immunosuperfinite. I think now there are about 30 labs in the world who are using different forms of the technology for their scientific research. So let's skip this part here. So next, I want to share with you on some unpublished work and some new development where we are trying to develop a sequence-based DNA nanoscope. So for everything I was shown here, talked so far was using DNA probes to visualize molecular structures or tissue structures. We need a microscope. We need a spatial mapping device which can essentially spatially map particular structures of interest. So in this new direction, we are hoping to develop capability where rather than using spatial mapping device that is using microscope, we are hoping to encode the spatial arrangement and also neighborhood proximity relation of individual molecular targets in DNA proximity records. Then by sequencing those DNA proximity records, we hope to reconstruct the local topology and geometry, and hence reconstruct the global geometry. And so, intuitively, what we do here could be very simple, right? You want to figure out the spatial arrangement of these three targets, red, blue, and orange. You can simply label this with three different DNA barcodes. Then if you have procedure to generate proximity records to encode red with green, and then red with blue and blue with orange, and by inspecting the red, blue, and blue-orange relationship, you can know the sequential arrangement of these three targets should be red, blue, and orange. So in fact, there's multiple different proximity ligation extension methods have been developed by the field. However, those methods are not suitable for this particular task. The reason because when you do proximity ligation to generate a proximity record, you will consume the underlying DNA record. That is, if the blue one is lucky to the red one, then it can no longer be lucky to the orange one. So that will give you only incomplete capturing or encoding of the molecular information on the single molecule scale. So to overcome this challenge, we invented a new molecular procedure we call auto-cyclic proximity recording, where we can generate the proximity records continuously and catalytically without perturbing the underlying DNA barcodes. And here's how it works on the detail level. So we use the two DNA hairpin species, PI and PG, and they encode the identity of the underlying target, I and G, but they share the identical universal and the primary binding region A. Then we introduce as a primer A star. A star can bind to A, and again through polymerase and polymerization, and A star can be extended to be J star and then later P star until it hits a stopper sequence. And similarly, we can also generate A, I, and A star, I star, and P star. Importantly, the P star, J star can compete with newly synthesized P star, J star of the PJ region, and this will avail it in the single-stranded version. Similar, this will also happen for this guy here. Now, the P star is engineered to be palindromic. That means it can bind to itself. So as a consequence, if these two probes are arranged nearby to each other, the two P star can bind to each other, and this will engage polymerase, which will stand on both sides to generate the full record. And importantly, it will strip the record away from the underlying hairpin species. Now we have the record encoding I and J star, but with the molecular barcodes regenerated, and you can go through multiple cycles and produce many different records. So this is animation to show the molecular process. We start with the two probes that bind to target A and B, and then you introduce primer. The primer will bind to the primer binding region. And the polymerase will be engaged, and you can copy the barcode until his stop region. Polymerase is displaced. Now the identity of target encoded in the half record. The newly synthesized half record is competed off the stem of the hairpin. Now, if A and B are sufficiently close to each other, this palindromic sequence can bind to each other. Polymerase will be engaged. Then we can synthesize the full record with the hairpins barcode A and B regenerated. So now you can do this over and over again to synthesize many different copies barcodes. So in here, then you can encode both these regions. So we demonstrate experimentally on a synthetic nanostructure where we have 7 different species and can generate proximity records. And by inspecting these proximity records, we were able to reconstruct the molecular topology with 30 nanometer resolution in this case here. So this is the first generation of so-called nanoscope we published a while back. And so I will demonstrate fundamental principle from a local proximity information inspection, we can reconstruct the global geometry. But the utility of the first version is quite limited, in the sense that the molecular logic here is really when two molecular targets are positioned below the threshold distance, then you can generate a proximity record. So this can work in the idealized dense packing configuration, but if you want to really apply it to the real and practically relevant context, for example, realizing biostructures, and this is not sufficient. So in the second generation, and a very talented postdoc in EQ, and combines the earlier idea of in situ PER single strand synthesis with the autocatalytic proximity recording, rather than generating a proximity record that really EQ extended proximity recording device into a molecular ruler where you can grow DNA polymers extensively in the space, in the two DNA strands meet each other, then you will, to the growing end meet each other, then you can synthesize full record. And importantly, these targets can be positioned at a longer distance, and the length of molecular record reflects the underlying physical distance. So after substantial development, EQ were able to show that, and with the sufficient statistics, and he can infer the length of the distance between two targets with a nanometer, 1 nanometer precision, and this again calibrated using DNA organic structure, where we can precisely position our barcodes at particular distance, then generate the records, and then through nanopore sequencing, and we measure the length and then fit the distance, fit the length to those underlying distance. So this offers quite an exciting prospect. So in a follow-up demonstration, and EQ demonstrate that, again here, this is still using synthetic nanostructure, but rather than 2 points, and he positioned the probes, and hundreds of probes with a very complex molecular geometry. And then by generating a population of DNA barcodes, where each DNA barcode encodes both identity of the underlying and the probe, but also importantly, the proximal distance of the length between the pairwise and the probes. And he sent this population of DNA barcodes to a handheld nanopore sequencer, then by inspecting the sequence information, and then he was able to reconstruct molecular geometry with a nanometer precision and accuracy. So shown here are the real data, not simulation. So here, the green dots indicate the design and precision for the targets, and the black circle actually is experimentally measured and computationally reconstructed precision of these molecular barcodes. And as you can see now, we can really visualize our sequence and identify the arrangement of this compact pattern. And this generalized to different molecular configurations. And this is quite interesting here. So here has 2 different patterns. And so because we can do longer distance measurement, and EQ was able to not only reconstruct the molecular arrangement of individual structure, but also their relative positioning on the in space. And since we are using sequencing rather than imaging to visualize, to identify these targets, and sequencing has intrinsic high multiplexing since each the targets is barcode indicates the identity of the underlying targets. And so we are looking at a very quite exciting prospect of a technology potentially have a 5 nanometer resolution and 100-plex and multiple capability. I say potentially because the technology, although this demonstration seems very, we are excited about where we are, but I should point out that the current technology is still not ready for primary and wide adoption yet, because we are still using ensemble data rather than the single structure sequencing data. And so next, Nikhil is hoping to turn this ensemble measurement into a single molecule measurement. Once we got there, I think that can be a very exciting prospect where we can potentially combine the genome-wide omic capability for sequencing and the spatial measurement offered by molecular imaging to offer spatial omics and the capabilities, for example, to visualize intracellular structure arrangement. Another very interesting high-value target could be to analyze the chromosome and genome architecture in the cell. Again, this is emphasized. This is a prospective possibilities. This is not experimentally de-risked yet. So I think I'm already kind of run out of time, so I'm going to maybe very quickly share some ongoing work we are doing on single molecule protein sequencing or protein fingerprinting. So we look at the technology development in the past few decades, and I think a number of things really stand out. One is sequencing, and then DNA sequencing, and this is really transforming biological research and transforming science, impact and transform clinical practice. And importantly, on the DNA sequencing, people not only can do very large-scale sequencing, the high accuracy, but people can go down to very high sensitivity in the sense that people can do single molecule protein and DNA and RNA sequencing. And this is in contrast with the protein sequencing or protein analysis. Although using targeted approach such as antibody and binding, and people can identify single molecule sensitivity proteins. But if you are talking about untargeted approach where we don't have affinity specific binders, and then we are restricted to mass spectrometry in practice, which is still a very powerful tool, which can identify in an untargeted fashion and the proteome of of cells. However, a key limitation of mass spectrometry is that it requires the millions of 1 million copy of protein to work robustly, and essentially that has a really much lower sensitivity compared with DNA sequencing. So recently there has been, I think, an exciting community effort trying to develop different ways to sequencing of fingerprinting proteins on the single molecule level without using any affinity reagent. And one approach we are taking is to utilizing our DNA capability. So the idea is that you take arbitrary protein and then you label particular selected amino acid residues with DNA barcodes, for example, lysine or cysteine. And then then you turn a globule protein into a DNA barcoded long polypeptide. Then through some imaging approach we have developed, and we hope to generate a particular optical signature of the DNA barcoded protein, and from the signature and from signature alone, we hope to identify what this protein is. And so there are really two kinds of signatures we are looking at. One is a geometric signature where you further stretch this DNA labeled polypeptide into approximately linear configuration. And then using the 5 nanometer DNA print imaging capability Mingji has published earlier, and he was hoping to compose, identify, and visualize the sequential arrangement of lysine cysteine along the peptide. And this signature, according to our analysis, should be sufficient to inform us what this protein is among the 20,000 different proteins in human protein database. And more specifically, according to our calculation, if we can have under ideal situation, just by visualizing lysine alone, we should be able to identify about 90% of human protein uniquely, ambiguously. But the other signature we are actually now focusing initially, instead of using the geometric signature, we are focusing on the amino acid counting, the so-called composition signature. The idea here is that turned out if you do analysis by only counting precisely how many copies of lysine cysteine are there in one particular protein, using these two residues, by counting their number, you can identify 2% of human protein. And then once you expand the 2 residues into 4 residues, and the coverage can go up to more than 70%. So a foundational capability for realize this vision is the new kind of geometry, some new chemistry Ming-Jie developed in the lab, where now he can label on the whole protein level, but this is not a synthetic or short peptide fragment. This is a native protein, and to label lysine and cysteine with 80–90% yield and efficiency. And this opens a possibility of precise signal identification based on DNA labeling. And on the counting side, we use another technology we developed in the lab called QPen, the counting pen. And this is a little DNA pen, but the key idea here is that rather than visualizing and distinguishing individual the precision different structures, and by looking at the image binding frequency and to the DNA barcodes, we can back out the number of underlying target without optically visualizing them. And this actually, the fundamental is very intuitive and simple. So you have 2 clusters here, and you have 1 binding set here, and 3 binding sets here. Then intuitively, the blinking frequency of this side will be 3 times more than this side. So then after development and optimization, we were able to achieve very high precision and accuracy for this blinking-based and quantification of molecular targets in the cluster. So then Ming-Jie applied QPen to the lysine labeled the whole protein. And then he was able to, for example, he look at a microscope, and this particular spot has a particular blinking kinetics. And then based on this kinetics, he was able to identify this particular protein is one of the 3 protein species they put together here. And then there's another guy. Now he further extended this to test 5 different proteins with a different lysine and lysine number. And quite excitingly, and he was able to show that the blinking signature, based on blinking signature, he was able to get a linear relationship of the measured lysine residue number versus actual lysine number. And this makes a method to be computationally and designable rather than empirically calibrated. So then without getting to, I think the time party is already up. So again, to summarize, so we work with DNA RNA molecules, and we engineer digitally precise molecular behavior and geometry dynamic synthesis of such digital information bearing molecules, and by interfacing such digitally programmable systems with other functional entities, and we hope to advance different aspects of practical applications and with imaging focus recently. So I want to acknowledge my many talented colleagues in the lab who made the work possible, our really amazing alumni, collaborators from the agency, and this is my disclosure. And I'll stop here to take some questions.
Thank you very much. I'll open it up for questions to the crowd. I have one in Q&A. If you have a mic and accessible, please come on. Go ahead, Julius.
Yeah, thanks, Peng. That was an amazing amount of work that you showed us there. I wondered, since nucleic acids are subject to a lot of design algorithms, as you know so well, and many of your technologies, have you developed computer-aided design tools to help people adapt this? And maybe you can talk a little bit more about the specific sequence design to implement these ideas.
Yeah, thanks so much and for the question, Julius. Yeah, this is a very important aspect, since the whole idea of this enterprise is that these things are designable, and the computing tools is critical. And so based on for different systems, we develop different design tools. And turned out the design requirement and the considerations for different systems are also quite different. For example, for the nanostructure and construction, where we designed hundreds of 10,000s of DNA break species to sub-assemble into 2D or 3D structure. And there, so key considerations is just to really design orthogonal sequences so that they can pair up with each other. And the very robust thermodynamic can drive these structures into particular structure, into particular form. So we designed a tool called NanoBrick. And this tool have a graphical interface where the user can essentially specify using a mouse to specify the 2D and 3D shape, and you click a button, and the sequence will be automatically generated, and you can send it to IDT chemical synthesizer to produce sequences. So for other things for other system dynamic systems, we also designed the more kinetic based software tools, and one is called the dynamic workbench, and which is also fully available. And for the application-based different application, we also design tools. For example, Saberfish, immune saber, these imaging tools, and we have different softwares that we share freely with the community, which you can download from the website. But we are just one small group in a very large thriving community, and there are many really excellent tools, including one developed by Julius and Young Lab to facilitate some design for DNA structure and applications.
Thanks, Peng.
Yes. Okay, go ahead.
This is Chad Merkin.
Hi, Chad.
Very nice talk, tour de force. Here, I have a more philosophical question. So you've developed incredible ways of making things that couldn't be made before. You've developed all sorts of clever and novel ways of detecting and monitoring things that were difficult to detect or monitor before. Are these techniques to the point where we've learned something we didn't already know? In other words, most of the way you present it is about doing things that we'd like to be able to do, but not a demonstration of learning something biologists didn't know prior to having these techniques. Are there examples yet, or is that still coming?
Yeah, thanks for the really excellent question, Chad. And these are very important considerations to think about what we want to do, how to design with the portfolio where we are. So, and I would say there are a number of considerations. And for the fundamental perspective, also we are really bio engineers or technologists. And so my lab has not been focused on mechanical study of biological phenomena. Although we do design tools to enable our study. On the fundamental side, I think there are a number of quite interesting things we learned about design principle for such synthetic nanostructures, and both through success and through tons of failures, and I did not have time to cover. And on the practical side, I think the tools we design are really, although we do not do too much biological discovery, these are intended to enable other people to do discovery. And I think there's a number of quite interesting things that has been visualized using both on the, for example, chromosome structure compaction using DNA-based DNA print tools for molecular scale imaging, and also certain molecular structures in biology on the molecular scale. And for the super imaging, for example, and this also enable people to visualize the particular patterns, in particular in the immuno-oncology context, to inform biology, inform the potentially drug design. So I would say some of the tools are still at a very early stage, conceptual stage, but some tools, especially dealing with imaging tools, at the stage really is already commercialized and to enable people to do better discovery, and also it's being adapted also by pharmaceutical industry to inform their patient stratification, drug design, clinical trial.
Okay, thank you.
Thank you.
Thank you very much for an excellent talk. I think in the interest of time,